Hello everyone, welcome to my YouTube channel Biologics by Sahana Gaonkar. In the last session we have studied about some basic aspects of transposable elements. So in today's session we'll study about Barbara McClintock's experiment. Let's get into the topic. Barbara McClintock's experiment. So in the 1950s, Barbara McClintock found a genetic element called DS, that is dissociation element, in the maize that causes a chromosome breakage at a point on the chromosome adjacent to its location. She also demonstrated that the phenotypic effect caused by the DS depend upon the presence of another element which she called as AC, that is activator molecule. McClintock found it is impossible to map AC and DS elements. They changed their position on the chromosome in different maize plants. In fact, dissociation element didn't just break chromosomes, but it could actually move from one chromosomal location to the another. Activator molecule is capable of autonomous movement, but dissociation element move only in the presence of AC. Insertion of AC element is unstable because it can direct its own transposition over and over. It can move independently. Insertion of DS, that is non-autonomous element, is stable in the absence of AC because it needs products of elements in order to transpose. So in maize, at least about 14 different transposable systems are known so far. So for example, EN element, which codes for single enzyme called transposase induced transposition, AC and DS controlling elements which encode transposase enzyme, MU, that is mutator element, and many more. Let's see what happens in Barbara McClintock's experiment. So this is the diagrams showing the transposon effect on the corn kernel color. So in the first diagram we can see that there is a activator molecule, dissociation element, and the normal C gene. So here there is no movement of activator molecule which activates the DS transposition. So there is no transposition takes place. So there is a normal expression of C gene. In the second diagram we can see that the activator molecule which activates the transposition of this dissociation element. So this dissociation element can transpose into a C that make the mutation or make the transposition. As a result, here the disruption of the normal C gene into a mutated C gene. As a result, we will get a yellow colored corn kernel. In the third diagram, we can see that the activator molecule which activates the DS element transposition out of the C gene a few cells during the kernel development. So this is also followed by reversion of mutated C gene into a normal C gene. But as a result, we will get a mosaic pattern because there is no 100% reversal of mutated C gene into a normal C gene. So we will get spotted corn kernel. Control of transposon. These are some important topics. Like control of transposon includes some autoregulation and transcriptional silencing. So some transposases are transcriptional repressors of their own promoters. Transcriptional silencing means mechanism which is not well understood but correlates with methylation of the promoter, also methylation of IRs. They do not produce phenotypic effect nor they actively move around the genome. These are some biological significance of transposon. So these transposons provide a means of genomic change and variation, particularly in response to stress. For example, there is a hypothesis called Barbara-McClintock's stress hypothesis. And also, these transposable elements played a significant role in the evolution of many genomes, as it involved in chromosomal variation and change. Also, as a genetic tools, DNA transposons can be used to induce a piece of foreign DNA into a genome, and there is no known example for an element playing a normal role in development. There is also one more topic called as transposon tagging. So in genetic engineering, transposon tagging is a process where transposons are amplified inside the biological cell by a tagging technique. So transposon tagging has been used in several species to isolate the genes. Also, it is used for insertional mutagenesis in the plants. For example, it has been used in glycine max. This is all about today's session. If you like the video, please share, subscribe, and thank you so much for watching.